Embryology, unveiling the intricacies of brain development. Welcome, medical students, to our comprehensive lecture on embryology, specifically focusing on the remarkable process of brain development. In this detailed script, we will delve into the complex phenomena that occur during embryogenesis, highlighting the significance of each stage in shaping the structure and function of the human brain. So, let us embark on this journey of exploration into the fascinating world of neural development. During early embryonic development, the neural tube forms through the process of neurulation. This intricate process begins with the formation of the neural plate, a specialized region of ectoderm. As the neural plate grows, its lateral edges elevate, forming neural folds. Eventually, the neural folds fuse together, initiating the closure of the neural tube. The neural tube acts as the foundation for the central nervous system, eventually developing into the brain and spinal cord. Any disruptions in neurulation can lead to severe developmental abnormalities, such as spina bifida or anencephaly. Thus, understanding the mechanisms involved in neurulation is crucial for preventing and managing these debilitating conditions. The neural crest is a group of special cells that migrate throughout the developing embryo. These cells are important for making the peripheral nervous system, as well as structures like the face, heart, and skin. Problems with these cells can cause birth defects. During early embryonic stages, the brain undergoes sequential formation and subdivision into the forebrain, midbrain, and hindbrain. These regions develop into specific structures that are vital for cognitive, sensory, motor, and autonomic functions. Disruptions in this process can result in neurological disorders and developmental delays. During early brain development, the ventricular system originates from the lumen of the neural tube. It plays a critical role in producing, circulating, and absorbing cerebrospinal fluid, CSF. CSF provides support, nutrients, and waste removal for the developing brain. Disturbances in ventricular system development can lead to hydrocephalus. During embryonic brain development, cells multiply to generate neurons and glial cells. Certain molecules and transcription factors help determine their fate and specialization. Glial cells have different functions, aiding in structural support, myelin formation, and immune response. Studying these processes helps understand neurodevelopmental disorders and potential treatments. Cellular migration is a sophisticated process that ensures proper placement of neurons and glial cells in the brain. This complex journey involves molecular pathways and guidance cues that guide neurons from the ventricular zone to specific cortical layers. Radioglia assist neuronal migration as structural support and guidance cues. Disruptions can cause structural abnormalities and impair brain circuitry, leading to neurodevelopmental disorders. Studying migration mechanisms offers valuable insights for understanding and treating such disorders. Axon guidance is a complex process in brain development that ensures proper connections between neurons. Axons navigate through complex environments using attractive and repulsive cues to reach their target destinations. Disruptions can lead to developmental conditions like dyslexia and autism. In the section on synaptogenesis, we will explore the remarkable process of synapse formation between neurons. Synapses are specialized junctions that allow for communication and signal transmission. This dynamic process involves complex cellular and molecular mechanisms, leading to the development of presynaptic terminals and postsynaptic dendritic spines. Many key players, such as cell adhesion molecules and synaptic organizers, guide the precise assembly and maturation of synapses. Understanding synaptogenesis is crucial for studying synaptic plasticity, learning, and memory formation, as well as for understanding neurodevelopmental disorders and neurodegenerative diseases. Myelination is a crucial process in brain development where myelin sheaths are formed around axons. These sheaths act as insulation and enhance the speed of neural signaling. Problems with myelination can cause neurological disorders. Studying myelination can help us understand these disorders and find ways to promote recovery. Apoptosis, or programmed cell death, shapes the developing brain by eliminating unnecessary neurons and establishing proper connections and circuits. Defects in apoptosis can contribute to neurological disorders. Studying apoptosis helps understand brain development and associated pathologies. Neurotrophic factors are growth factors that guide the survival, proliferation, and differentiation of neurons during brain development. They include NGF, BDNF, and GDNF. Disruptions in their availability or signaling can lead to neurodevelopmental disorders and neurodegenerative diseases. Studying neurotrophic factors can inform therapeutic interventions for neurological conditions. During critical periods of brain development, the brain is highly adaptable and can be modified based on experiences. These periods are important for sensory perception, language learning, and social development. Environmental factors greatly influence brain development during these critical periods and can have long-term effects on cognition and behavior. Providing enriching environments and timely interventions is crucial for optimal brain development. Embryology sheds light on brain development, from cell processes to functional circuits. Factors like myelination, apoptosis, and neurotrophic factors contribute to brain complexity. Understanding genetic and environmental influences aids in neurodevelopmental disorder research and patient care. Embrace this knowledge for a successful medical career.